Okay guys, welcome back to part 2 of the video lecture for chapter 12. We're going to wrap this up with this second part here. So you remember at the end of part 1 we had just sort of finished talking about the overall process of mitosis, which is division of the nucleus. Gone through the various phases of it: prophase, metaphase, anaphase, telophase, all that. Next thing to talk about is cytokinesis. Now this is a process that occurs usually begins toward the end of anaphase or starts in telophase, kind of varies. But this is the process that divides the nucleus, or separates the cytoplasm rather, between the two nuclei, so that you end up with two cells that both have a copy of that nucleus. Mitosis is division of the nucleus. Cytokinesis is division of the cytoplasm between them that results in the cell splitting in two. Okay. So this happens in one way in plant cells and another way in animal cells. And the reason for the difference is because plant cells have a cell wall and animal cells don't. The plant cell wall is very rigid, okay? So let's look at animal cells first. On the left-hand side here you see what's called a cleavage furrow of an animal cell. That's the cleavage furrow right there. What's happening is that on the inside of the cell membrane between the two nuclei, there's a circular ring of contractile proteins, microfilaments, one of those three protein fiber types we talked about that make up the cytoskeleton. These act like a drawstring. They cinch down to a central point, and the two membranes of the two cells separate, and now we end up with two identical daughter cells, okay? That's cytokinesis in animal cells, basically. In plant cells, again, that contractile ring of proteins, if it existed, would not cinch down those cell walls because the cell walls are rigid. They're not flexible and flimsy like the cell membrane. So what happens is that vesicles from this side of the cell and this side move toward the center, and these vesicles contain cell wall building materials like cellulose and various other things, and they begin to collect and fuse together in the middle, right here. And as they begin to fuse, they start to build a structure called the cell plate. It's like a sac. And as more and more vesicles fuse with it, it gets bigger and bigger until it eventually contacts the existing cell membrane and cell wall and separates those two nuclei from each other. So you can think of this process of cell plate formation in a plant cell, in other words, how they carry out cytokinesis, as a process of breaking up a new cell wall between those two nuclei. Okay, and again, that has to happen because the cell wall is rigid and it won't cinch down like the cell membrane of an animal cell will during cytokinesis. Okay, so I have a video to kind of show you the differences then between animal and plant cell cytokinesis.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Okay, so now let's kind of put all this together. Let's look at some actual photos of cells going through the various stages of mitosis and cytokinesis, okay? These are our photos of what are called onion root tips, okay? So cells of onion root tips. So if you go to the grocery store and you buy a bundle of green onions, for example, they very often have that sort of whiskery kind of structures hanging off the end of the onion bulb. Those are roots. And so if you look at the tips, that's where cells are actively dividing, and you can see cells in various stages of mitosis, and that's what you're seeing here. So first what we have is a cell in prophase. All right, so what you're seeing here is a circular nucleus, but notice how it's very dark and irregular in coloration, very grainy in appearance. That's a dead giveaway that you've got a prophase cell. Compared to the interphase cells next to it or up here where you have a nucleus that's more even in coloration, all right? Here we have a late prophase, again what the author calls prometaphase, just call it late prophase. Now you can start to make out individual strands, thick strands of chromosomes. So this is a later stage than this earlier prophase, all right? Nuclear membrane is starting to break down, but you can't really tell that from the photo. All right, third we have metaphase down at the very bottom, and now you can sort of tell there's a line here where the centromeres are sort of lined up. You can see that, and that's that metaphase plate we talked about, that imaginary two-dimensional plane. Nuclear membrane is broken down completely, it's gone, and all you see are the chromosomes, but they're all sort of lined up on that imaginary two-dimensional plane. All right, anaphase. This is not a very good anaphase photo, but you can see one set of chromosomes sort of here basically, and another set there. You can see a little bit of space in between them. Those are that's an anaphase cell. You start to see space between two distinct sets of chromosomes as they're moving toward opposite poles of the cell. And then finally, and this is actually a really good telophase here, you can see clearly two very distinct nuclei, and those chromosomes in those nuclei are now starting to decondense back into chromatin, but you can also see a very faint cell wall forming there. Hasn't quite connected on this side or that side yet, but that's a telophase and also a cytokinesis simultaneously. So that cell plate is that new cell wall being built to separate those nuclei from each other into separate cells. All right, so next thing to talk about is the system that basically tells a cell when to go through cell division. So we have a system called the cell cycle control system, and this basically regulates when a cell goes into cell division. It basically regulates the cell transitioning from one part of the cell cycle to the next. For example, G1 to S to G2, into the mitotic phase, and so on. So the frequency of cell division varies with the type of cell that we're talking about. Skin cells and blood cells divide very rapidly. Skin cells divide very rapidly. For example, bone cells don't at all. Well, they do divide, but not very rapidly. Nerve cells, once you're fully grown, don't divide at all. So there's this control system that regulates how often cells divide. And one of the reasons why there's so much research funding being poured into understanding this cycle is that we know that cancer is a result of a malfunctioning cell cycle control system. So in a bid to ultimately cure cancer, we have to understand why it's occurring, and we think it's a result of some aspect of the cell cycle control system not working properly, okay? Because cancer cells manage to escape the usual controls on the cell cycle put in place by the control system. All right, so the cell cycle appears to be driven by specific chemical signals present in the cytoplasm. Typically, these are proteins, as you might expect, and we're going to talk about some of those here in a little bit. But the events of the cell cycle are directed by a distinct control system, which is sort of similar to a clock, okay? The control system is regulated by both internal and external controls. And I'll explain what I mean by that here in a little bit. But it has also specific checkpoints where the cell cycle sort of pauses until a go-ahead signal is received, okay? So if we lay that control system over the cell cycle, this is basically what we see. All right, so again, we have from this point, we have interphase G1, S, G2, all the way around. Then we have the mitotic phase here, so mitosis, cytokinesis, okay? There are 3 checkpoints. We have what's called G1 checkpoint there. We have G2 checkpoint at the end of G2, and then the M checkpoint. These are periods of time when the cell cycle basically pauses, okay? You can think of them as sort of, in particular, the G1 checkpoint as sort of a gut check, right? The cell kind of pauses and says, okay, is everything that's gone on to this point happened correctly? Are we okay? Are we good to go forward, all right? Turns out G1 checkpoint is the only checkpoint that can cause a cell not to divide. In other words, that's the only checkpoint that can stop the cell cycle. The G2 checkpoint and M checkpoint down here are really just pauses to allow time to make sure certain things have happened, okay? So the G1 checkpoint seems to be the most important one because it's the only one that can stop a cell from actually dividing. All right, So there are a number of signals that are registered at these checkpoints that come from the cellular sort of surveillance mechanisms within the cell, okay? And we're going to talk about some of those here. So let's assume that the cell goes into the G1 checkpoint and it receives a go-ahead signal, okay? That means it goes on into S phase where it copies its DNA, goes on into G2 where it copies the organelles, starts building the cellular machinery it's going to need for mitotic phase, and then it goes on through mitosis, cytokinesis, and divides in two, okay? Now, what if cell goes to G1 and there's a problem, and the cell gets a signal to stop the cell cycle? Well, then that cell will go into a phase called G0. Okay, G0. That's not go, that's G0. G0 phase. This is a non-dividing state. Okay, you can think of it as sort of like a state of suspended animation. That cell will go into G0 phase, and basically it goes through a process whereby it ends up self-destructing. That process of self-destruction is called apoptosis. That second P is silent, apoptosis, all right? This is also referred to as programmed cell death, okay? Now, this is important because, as I said, in this example or this scenario, you have a cell that has a major problem. Maybe it has an extra chromosome or some other problem with the DNA, who knows? You don't want that cell to go on and divide, because if it does, it could potentially pass on that problem to every cell that descends from it. And that could be hundreds or thousands of cells, at some point in the future. All right, so this programmed cell death, apoptosis, gets rid of that cell. Basically what happens is that when the cell goes into G0, the DNA of the cell fragments and breaks up, and that causes the cell to die. Okay, and the cell then fragments, and then the body sort of recycles all the components of the cell. But this process of apoptosis is also an important part of development. At certain stages during fetal development, okay, organisms, animals in particular, will have certain structures form that then get either pared down or removed entirely. So for example, every human that's on the planet today had a tail at one point during fetal development, but we're typically not born with that. That's because that tail structure is destroyed through the process of apoptosis as embryological development goes forward. In addition, we all had webbing between our fingers and toes, but we're not born with it, usually. And so that's because apoptosis destroys those cells of the webbing before we're actually born. All right, it's just a normal part of development, okay? All right, so let's take a look at this process then of these checkpoints coming into play. So in our first panel, panel A up here, we have a cell that's reached the G1 checkpoint and it's received a stop signal. There's a major problem. Cell goes into G0 phase, and that's when apoptosis then occurs to destroy that cell, okay? It could be that there's not a problem. It could be it's a cell that belongs, or that's part of the webbing or the tail that we have during certain stages of development, and it needs to be destroyed as a part of getting rid of that structure, okay? All right, now, in in the next step over here, let's assume that we have a cell that goes on through the G1 checkpoint, goes through it, it will then go on and divide. All the rest of the cell cycle will take place as usual, okay? All right, well then, what are G2 and the M checkpoints about? Well, the G2 checkpoint is basically a pause where the cell kind of stops and says, okay, was all the DNA that was copied during S phase, was that process completed? Was DNA replication completed? You don't want the cell to go on into mitosis if it hasn't yet finished copying its DNA. That means one or both cells might end up missing some DNA, and that's usually going to be a death sentence for the cell. So G2 in particular seems to make sure there's time for all the DNA to be copied, but also all the organelles to be copied, which is what's happening in G2 as well. Okay. All right. Then cell goes through G2, it encounters the M checkpoint. So what's going on with the M checkpoint? Well, main thing there is that, if you remember, we have in mitosis chromosomes lined up in the center of the cell. You got a centrosome there, and your fibers are growing out and connecting to those chromosomes, as we said. But those fibers don't all grow at the same rate. So let's assume, for example, that we have a fiber, like I've shown here, that's not quite yet connected to the centromere of that cell, the kinetochore of that centromere. If that cell went ahead on into anaphase, this entire chromosome would get pulled that way, right? Whereas these centromeres would split, chromatids separate like normal, but because that fiber is not connected yet, that chromatid there would end up going this way. That is something called non-disjunction, meaning that those chromatids did not separate. And that would mean this cell would end up with an extra copy of that chromosome. You might think, well, why would that be a big deal with extra DNA, a duplicate copy of a chromosome? Well, it can be a big deal. Probably heard of Down syndrome. We all know when we see somebody who has Down syndrome, because they have the very distinctive facial characteristics and so on, Down syndrome is also called trisomy 21. Somebody with Down syndrome has 3 copies of chromosome 21 instead of the normal 2. That's a result of a non-disjunction event that occurred probably during meiosis, gamete production, which we'll see in the next chapter. But this is just an example of how chromosome number being incorrect can cause serious problems. Whether you're missing an entire chromosome or you have an extra one, it can cause problems. So that M checkpoint is to make sure to give time to allow all those fibers to properly connect to all those centromeres. Okay, so that's an example of an internal signal. Okay, you have a signal from inside the cell where a fiber is not yet connected. That's an internal signal that would cause a cell to wait, rather, at the M checkpoint. All right. What about an external signal? We'll think about growth hormone. Growth hormone is a protein that's produced by the pituitary gland, and as we're growing, from birth to 15, 16, 17 years old, whatever, we have higher levels of growth hormone in our bloodstream than we do as adults that are no longer growing. The higher levels represent an external signal, something outside the cell, a hormone that tells the cell to divide at higher rates. So you're putting on more cells on a daily basis than you're losing, and so your body grows and gets bigger. At maturity, when we stop growing, our growth hormone levels drop so that we're just putting on enough cells to replace the cells we lose on a daily basis. That's why the body stops growing once you reach maturity. But growth hormone is an example of an external signal that causes cells to divide. Another example, wound healing. Let's say you have a cut or a burn or a broken bone or something. The process of wound healing is an accelerated rate of cell division. That's an external signal that causes cells to divide at higher rates. For example, you can take and grow cells in a petri dish, and those cells will cover the bottom of that dish, and once the cells have reached the walls of the dish and they all become crowded together, that crowding is an external signal that causes them to stop dividing. Then you go in and scoop out some cells to simulate a wound, and the cells that border that scooped out area will then begin to divide because they're no longer crowded. You don't have cells contacting them on all sides, and they'll grow in together. That's called contact inhibition. So contact inhibition is another example of an external signal that causes cells to stop dividing once they're contacted on all sides by other cells. A burn or a cut or something like that destroys some cells. The live cells next to them are no longer contacted by live cells. They begin to divide again to sort of fill in that wound or heal that wound, so to speak. Okay, so that basically wraps up chapter 12 on the cell cycle, mitosis, and cytokinesis. On the next chapter, chapter 13, we'll talk about a more specialized type of cell division called meiosis that produces just sperm and egg. Okay? All right, let's wrap this up.